Hello Bruins, this is Ms. Farouk again, and this is part 2, continuing with part 1 with translation. Sorry that video stopped suddenly. And so continuing from there, we were discussing about translation. So the major players in translation is shown in this picture. So here you have your ribosomes, and this is where your mRNA that has the instructions to make protein resides here at the ribosome, and then the tRNA will come in and match up, recognize that codon, and bring that complementary amino acid to assemble the polypeptide. Now remember, translation means protein synthesis, so we're now going from mRNA to protein. So to begin translation, you first of all start with your mRNA, and on the mRNA you have the start codon, AUG. And then AUG is going to be recognized by a tRNA that will have UAC, which is called the anticodon. And this anticodon is going to bring the complementary amino acid called MET. And then we add energy. GTP is just like ATP in terms of using it as energy, and that helps to assemble this translation initiation complex by adding this large ribosomal subunit. Now if you notice, there's 3 sites on the ribosomes. There's E, P, A. And A is going to be the site where the tRNA will come in, and then when another tRNA comes in, it will then move to the P site, and think of P site as a growing polypeptide chain. And then another tRNA will come in, and as it's coming in, this bond will be broken from the tRNA, and then this amino acid will form a peptide bond with the next amino acid. And then this will move along to the E site. And then think of E site as the exit site, and that's the site where the tRNA is going to exit. So this is how it elongates. So here's your ribosome, here's your first step. You've got the MET already there. The next tRNA will come in, and notice this bond will from this tRNA will break and form a peptide bond with the next amino acid. So this peptide bond forms, right? And as you notice, the growing tRNA goes to the next tRNA, and then this tRNA will exit and wait for the next tRNA to come in. So it's like this factory. And then it keeps doing that until it reaches a stop codon, and these are the 3 stop codons. And when it reaches a stop codon, there's a release factor that binds to this codon, and when it binds, then the whole translation complex is released and disassembled. It releases this free polypeptide, which will then eventually go to the ER or Golgi body to get further chemical modification to become a functional protein. And then the ribosome unit will disassemble. The mRNA will probably be disintegrated by enzymes, and then that's the end of translation. Okay, so let's start with transcription again. So how do you get from DNA to protein? So first of all, in the nucleus you have your DNA right here, and then you gotta first make mRNA. So you gotta first do transcription. So the major players in transcription is your RNA polymerase. That is the enzyme that will add the RNA nucleotides. And here on the DNA, you have a promoter. It's a sequence of nucleotides where the RNA polymerase knows this is the beginning of the gene that's going to transcribe. Okay. So your RNA polymerase will bind to the promoter and add complementary nucleotides, and this mRNA is growing at the 5' to 3' direction. And then when it reaches a sequence that's called a terminator, it knows that's where the gene will end. This is where the mRNA knows when to stop, and it releases from the DNA and mRNA. So now you have a transcript of mRNA, right? And now it's ready to go out of the nucleus and then go to the ribosome. Now notice, remember, in eukaryotes there is an additional step, but to simplify things, we're just going to take now this mRNA to the ribosome. Now, once it goes out of the ribosome to the cytoplasm, it is ready to translate or do protein synthesis. So, what do you need for protein synthesis? Here you have your mRNA with the codons. You've got tRNAs with its anticodons, and you got amino acids, and here's your ribosome. So the first is the initiation step. Here's your mRNA. It binds to the small ribosomal subunit. And the first tRNA is your start codon, and your tRNA with the MET will recognize that start codon, and once that binds, then the larger subunit will come and bind, and that forms the translation initiation complex. Now you're ready to assemble your amino acid. Okay. Now this lysine will come into the A site, and again it recognizes that codon, and then the previous tRNA gives that polypeptide to the coming in tRNA, right, and then exits at the E site. Now the next tRNA will come in, and this will happen again, and now the polypeptide chain is transferred to the coming in tRNA, and then the tRNA deposits its amino acid and then goes to the E site, and then the next one comes in. So this keeps going, and as you can see, this is how the amino acid chain is growing, or polypeptide chain. Okay. And think of this as all as an enzyme, and the ribosome, or should I say ribozyme, is all breaking these bonds, forming these bonds, and an enzyme, as an enzyme. Now the releasing factor will come and bind into the stop codon. So here is one of the stop codons, and the releasing factor will bind to it. Now it knows this is the end of the translation, and once that happens, everything is released. Thanks for listening, and if you have any questions, please ask in class.